Hey everyone, in this video I'm going to be talking about changes in the different structures of DNA throughout the course of mitosis. So I know that this is going to be mitosis because I have G1S, G2, then I have my M phase followed by cytokinesis, but there's not a second M phase that follows after it. So since this is one round of division, we are looking at mitosis. Okay, so on my y-axis, I have 0, 1x, and 2x. And we're going to think of 1x as the normal amount. 2x would be double the amount, and then 0 would be 0. And we're going to start by looking at just the changes in the amount of DNA. I'm going to be looking at DNA, and I'm going to be using this red color here to show the changes. So at the beginning of G1, we would expect any of the cells, the daughter cells, to have the normal amount of DNA depending on the number of chromosomes. It doesn't matter how much it is, but we're expecting the normal amount. Then during the S phase, we would expect to see double the amount of DNA because the DNA has been copied. And once that duplication happens, we're not going to cut the amount of DNA in half until cytokinesis. So as we look at the changes in the amount of DNA over the course of the cell cycle, our graph is going to look something like this. Next, we're going to look at the number of chromosomes. And starting in G1 again, what we would expect to find is 0. We should not see any chromosomes during G1 or during interphase because at this point the cell has its DNA as chromatin and the chromosomes don't begin to form until prophase. Now during metaphase, the number of chromosomes should still be the normal amount. But during anaphase, we're going to see the number of chromosomes double because we are splitting the sister chromatids into their individual chromatids, and each one of those has a centromere. And once again, we wouldn't see the number of chromosomes go back to normal until we get to cytokinesis. And then at that point, it will continue to decrease until all the chromosomes have unraveled back into chromatin. So you can already see a pattern on this graph where talking about two different structures in terms of the DNA are going to have different implications. So if you're talking about simply the amount of DNA, then you are looking at the graph in this format. But if you're talking about the number of chromosomes, you're looking at a completely different set of data points based on how we define what a chromosome versus what we define simply as DNA is. So make sure you're on the lookout for the distinction between those two different types.